It was very encouraging. I would say it's going according to plan. This is what was expected from the previous trials. Having both antibody and T-cell response is clearly good, which is better than some of the other vaccines that are in development that are just getting antibody response. That said, the immune system is very complex and it requires more than simple measures to get there. So I'm really encouraged by this news. It shows that things are progressing. The real moment comes, though, in the pivotal phase 3 trials, because that's when you link it much more closely to the efficacy with patients and specific patient cohorts. There is some indication that the dose may need to be increased. And of course, when you increase the dose, you have trouble with safety. So there's still a balancing act. And I think the public is getting used to clinical development, which is risky and has moments through it that you get encouragement, but can get disappointment later. So I would say tempered optimism. It's great to see them there. What I'm also very encouraged by is that they're not the only one. There are 23 candidates now in clinical trials, which is a huge increase when you consider there were none only a few months ago. And that's a marvelous reflection on the industry.
Certainly, yeah, it's pretty tremendous when you think about what's been done by the healthcare community. So dosing is one thing to look out for when it comes to the future of the University of Oxford AstraZeneca vaccine. You mentioned the other vaccines that are in development. And yesterday, right alongside the Astra data, we also got some data on CanSino Biologic, a vaccine they are producing that uses the same method that is being employed by University of Oxford and AstraZeneca. And the results seem to be pretty similar. So my question to you, when we get data like this for a type of vaccine that is similar to others, so in this case viral vector vaccines, or in the case of Moderna and Pfizer and BioNTech messenger RNA, is it likely that one bucket of vaccines is going to prove effective? In other words, if we get one piece of positive data for one type of vaccine, should we extrapolate that to others that are using similar technology?
I think the technology platform is, of course, an interesting point, and you should be encouraged when two using the similar technology platforms have similar results. That gives further validation of the technology. I think the question really regarding all of the platforms is whether you're going to need a combination of vaccines here. It may well be that one works particularly well in a certain patient cohort and another one works particularly well with another patient cohort.